Good evening, everyone. It's so great to see you this evening, although with these bright lights, it's a little hard to see everybody, but it's great to have you all here. I'm glad the weather cooperated because it was pouring rain this morning and we were lucky tonight. I also want to welcome the live stream audience. I don't know how many people are online right now, but I'm looking forward to finding out. My name is Amy Harris, and I'm director of the University of Michigan Museum of Natural History, and we're the presenter of this program. For those who are wondering, this lecture is being recorded and will be shared later via the museum's website and our email museum at home that will be going out later this spring. Before we get started, I'd like to acknowledge that the University of Michigan is located on the territory of the Anishinaabe people. In 1817, the Ojibwe, Odawa, and Potawatomi nations made the largest single land transfer to the University of Michigan, seated in the treaty at the foot of the rapids. We acknowledge the history of Native displacement that allowed the University of Michigan to be founded. Today, we reaffirm contemporary and ancestral Anishinaabeg ties to the land and their profound contributions to this institution. The Museum of Natural History acknowledges the complicated history of settler colonialism and recognizes indigenous sovereignty, the ongoing effects of colonialization, and the global struggle for self-determination of indigenous communities. Tonight's program is the 23rd annual William R. Farrand Memorial Lecture. Since it was established in 2000, we have enjoyed a wide variety of topics that you can see listed in your program. The series is supported by an endowment funded by generous donors who you can also see listed in your program. Thank you so much for your gifts in the past and to those who have continued to give. Anyone can make a gift to the Farrand Lecture at any time via the Leaders and Best giving page on the University of Michigan website. The annual lecture was established in honor of William R. Farrand, Professor of Geological Sciences and Director of the Museum of Natural History from 1993 to 2000 under its former name, the Exhibit Museum. Sadly, Bill passed away in 2011 and the lecture became a memorial lecture. Tonight we're so happy to have with us Bill's daughter, Michelle Stearns, and baby William, he's just 3 months old. He is visible right here in the front. We know that Bill would be so pleased and proud to know that his grandson is here and that he's named for him. And I also want to recognize Ben, Will's dad. So we're really glad to have you with us tonight. Before we proceed with our program, please silence your cell phones. And now I'd like to introduce our moderator for tonight's panel discussion, April Baer, host of Michigan Radio's Stateside program. She will introduce the panelists and lead a moderated Q&A with the audience after their presentations. April, thank you so much for moderating tonight.
Thank you, Amy. Thank you, Amy. I'm not sure why I'm leaning into that because they also rigged me up with a mic tonight. I was very excited when I was asked to be here tonight. I'm a recovering arts reporter who has a special interest in the intersection between the arts and the sciences. And I could not be more thrilled about the work that these three panelists have done. The subject, of course, is microbes. Can any of us really say, can any of us remember a decade when microbes were not more a part of our life than they have been recently? Personally, I looked forward to this as an opportunity to get back in right relation with microbes. It feels like the moment to do that, and I can't think of three people better suited for this. I think, can I introduce you to them as a whole, and then they'll just come up and talk to you about their work one by one? Does that sound all right? Okay, great. Our first presenter is Anthony Vecchiarelli. Did I get that right? I asked him everything except how he says his name. He's an assistant professor of molecular, cellular, and developmental biology here at the U of M. And his lab uses a variety of disciplines, including but not limited to genetics, biochemistry, and cell biology to better understand how bacteria organize the fundamental processes that allow them to exist in cells. You might have seen his research downstairs over at the museum as part of algae and the climate. It's on algae and the climate crisis as part of the wider exhibition in the basement. Erica Lee Gardner is here as well. In addition to being an exceptionally snappy dresser, she is a PhD student at the Department of Chemical Engineering and is the creator of the Microbial Masterpieces project, which aims to communicate to all of us the importance of microorganisms to humanity. I mean, we could probably open that up on a global to all life on the planet too, but I'll let her get into that a bit. And she's also the originator of a visually stunning display of microbial life that is sort of the keystone work of Microbial Masterpieces. Also, you can also see that next door if you have not already. It's just, I could have stood there for an hour. Jim Cogswell is here too. He's an artist whose studio practice encompasses a variety of 2D and 3D media, painting and drawing and printmaking and design processes, and he's also on faculty at the Stamps School of Art and Design. He's a frequent collaborator with other artists and scientists across a variety of disciplines. If you're having thoughts as they're talking, please do feel free to call for pen and paper and write things down, or just jot something down on a piece of paper that you may have. We're excited about your questions. Some people have already suggested some questions online, and we will be sure to get to those as well. Right now, I'm going to turn it over to Anthony Vecchiarelli.
Okay, thank you. Today we're going to introduce you to a world that captivates us, the world of microbes. Our emphasis is on the natural world, how it can be profoundly influenced by a world that we can't see. The focus of the exhibit that I built in the People and the Planet gallery is about photosynthetic bacteria that can sequester carbon dioxide from our atmosphere and convert it to sugar, which is then used by the cell to grow and reproduce, just like plant photosynthesis. And so my exhibit also highlights a compartment that's in these bacteria that are not found in plants, called the carboxysome. I like to call it the most important organelle that most people, even scientists, have never heard of. This compartment is what endows bacteria the ability to sequester and convert carbon dioxide into sugar at much faster rates than plants. But first, I'd like to step back and first ask you the question, what do you think of when you think microbes, like viruses, bacteria, and fungi? The usual responses involve things like infection, sickness, contamination, unsanitary conditions, decay, and of course, real as well as currently fictitious pandemics. As a result of this thinking, we view our relationship with microbes as one where we have to get rid of them at all costs, brushing our teeth to remove bacterial biofilms that we call plaque. Brush your teeth, I'm not saying. Treating infection with the use of antibiotics, sanitizing, disinfecting surfaces we touch as well as the hands themselves, and preventing microbial transfer through the use of gloves and masks, things that we're all too familiar with nowadays. We are all now hyper aware of the microbes that can harm or even kill us. But the vast majority of microbes living in us and on us are not pathogenic and are actually very important for our health. We have roughly an equal number of microbial cells living in us and on us than we do human cells. So we are just as microbial as we are human. And this sentiment and its ramifications for human health are only now being appreciated by scientists and hopefully the public. Microbiome research is showing how pre- and probiotics aid in digestion, prevent gut issues, ward off pathogens, train and strengthen our immune system, and also how they can control our mood as well as our behaviors. But my lab focuses on the importance of microbes in a broader context. How are they important for the health of our planet? Without microbes, all life on Earth would collapse. These thought experiments have actually been done. Imagine wiping out every microbe on Earth. You may initially think, sweet, no more infections, the ideal world for germaphobes. You would actually be able to do surgery in a barn and you wouldn't get an infection. No need for antibiotics. Happy days, right? But we also won't be able to make beer or wine. That's a juice box. And more seriously, whatever food we had, we'd have a very difficult time digesting without our gut microbes, and we'd quickly suffer from malnutrition. What would happen to the Earth without microbes? That's the real horror story. Plants require nutrients in the soil that are made by microbes. As a result, crops will die off, we'll have massive food shortages. Even cattle and sheep rely on bacteria in their gut to break down all the plants that they eat, and they too would die from malnutrition. But it's really the lakes, the rivers, and the oceans where the global catastrophe would be most severe. Most microbes are critical for our water ecosystems. Without them, all fish would die. In short, without microbes, everything dies. But it actually gets even more sinister than that. Without microbes, there would be no decay. So in just a few years, if we survived that long, we'd be knee-deep in animal corpses and leaf litter. So the world would essentially become a graveyard. It gets better eventually. I just gotta finish this thought experiment. If that weren't bad enough, microbes produce a lot of oxygen through photosynthesis. In fact, the oxygen in every one of the 3 breaths that you take comes from microbes in our marine ecosystems. So what's shown here in this beautiful satellite imagery are the 4 globally dominant types of microbes responsible for making oxygen. We work on one of them in our lab called Synechococcus, shown in cyan, dominating the middle half of the Earth. Without these microbes, we'd watch the world die, and then we'd suffocate to death. We need microbes for human health. We need microbes for the health of our planet. And so this is what brings me to my exhibit called Algae and the Climate Crisis. We have put too much CO2 in the air, which acts as a blanket. It traps heat that would otherwise radiate into space. As a result, we're seeing extreme weather, these massive algal blooms across Earth's marine and freshwater ecosystems. And although we call them blue-green algae, they're actually cyanobacteria. And just to give you a sense of scale for these algal blooms that you could see from space, if you can see those little blips of color that I've circled, those are actually oil tankers, just to give you a sense of scale for these algal blooms. So these cyanobacteria, they created the first oxygen on Earth 2.5 billion years ago. They were responsible for the Great Oxygenation Event. They are responsible for cooling the planet, and they're the ones that allowed our oxygen-breathing ancestors to evolve. And so the question that my lab is trying to answer is, can they help us combat climate change, and if so, how? So land plants are well known to sequester atmospheric CO2, but they only do half of the work. The unsung heroes of the other 50% are photosynthetic microbes like cyanobacteria. Although plants and photosynthetic microbes can convert carbon dioxide to sugar, my exhibit explains how bacteria actually do it better. So both plants and photosynthetic microbes contain an enzyme that's shown here called ribisco. It's the most abundant enzyme on Earth. And ribisco is the key enzyme that converts carbon dioxide into sugar so that cells can grow via photosynthesis. But luckily for us, one of the byproducts of photosynthesis is oxygen. But that's a problem for this enzyme. It doesn't do a very good job distinguishing between carbon dioxide and oxygen. It can bind to either. And if it does bind to oxygen, it goes through this wasteful side process called photorespiration instead of photosynthesis. And so it turns out that evolution used 2 very different solutions to solve this oxygen issue in plants and cyanobacteria. So plant ribisco evolved to bind carbon dioxide better than oxygen. But that came at a cost. Tighter binding to your substrate means that you have a lower activity. Bacterial ribisco, on the other hand, was able to maintain its high activity, 150 times more efficient than plants at converting CO2 into sugar. But oxygen is still a major competitor for this enzyme. So How did cyanobacteria solve this oxygen issue? Well, it turns out that cyanobacteria hide their ribisco enzyme from oxygen in a protein shell called the carboxysome. So you can see inside this cyanobacterial cell how these carboxysomes are those little black blobs that are shown by the black arrow. But if you purify these carboxysomes away from the cell, you can see that they have an icosahedral structure. They sort of look like viral capsids, but instead of having genetic information within, they're chock full of this enzyme ribisco. And so the carboxysome has this shell made up of thousands of proteins that tile together to form the compartment. And if you look inside the shell, you'll see it's jam-packed with ribisco enzymes. And so in the People on the Planet exhibit, we constructed 3 massive and interactive carboxysomes to explain the 3 reasons why carboxysomes are so good at converting carbon dioxide into sugar. It has a protein shell that allows only carbon dioxide and not oxygen in. They have a densely packed enzymatic core. And finally, a highly efficient chemical reaction within. So let's go through those 3. The shell has special pores that lock out oxygen. Visitors can turn the carbon dioxide key to unlock the riches inside the carboxysome. This selectively permeable shell creates a treasure trove of carbon dioxide inside carboxysomes, making ribisco enzyme activity way more efficient in turning CO2 to sugar. The icosahedral shape actually fits the greatest number of ribisco enzymes in a tiny space, so there are more ribiscos to do the work. And so concentrating carbon dioxide around tightly packed ribisco is the reason why carboxysomes can process carbon dioxide so efficiently. And finally, the carboxysome's chemical reaction works sort of like a gumball machine. It uses carbon dioxide as currency to get the sugar cyanobacteria need to grow. So visitors can turn the dial, insert CO2 coins into the carboxysome candy machine so that they can be converted into sugar. So in my lab, many of which are here today, thank you for coming, y'all. These amazing graduate students and postdocs research how carboxysomes assemble, how they are organized in the cell, and how they function in the cell. And so why is this important? Well, I hope many of you are appreciating now how atmospheric CO2 has been climbing since the beginning of the industrial era. We reached the highest point in the last 2 million years. We're all the way up at the top right of that graph now. You can see now almost daily how climate change has become extreme, catastrophic, and almost irreversible. So when I started my lab, I chose consciously to work on carboxysomes for this reason. How can we use cyanobacteria and carboxysomes to combat climate change? Well, for example, cyanobacteria are being tested as an effective way to capture carbon from coal-fired power plants. Vertical bioreactors of basically PVC piping grow cyanobacteria that's exposed to sunlight and can capture carbon dioxide for photosynthetic growth. And then that cyanobacteria can be then concentrated to be used for biofuels or feed. But ultimately, more than one carbon dioxide source got us into this mess. It will take more than one solution to get us out. And so in collaboration with the Global CO2 Initiative here at the University of Michigan, we end the exhibit by highlighting the myriad commercial products now made using sequestered CO2, hand sanitizer, coasters, concrete, crayons, thereby potentially turning a liability into an opportunity. What else can we do with carboxysomes? Well, with our population expected to reach roughly 10 billion by 2050, there is increased pressure to make our crops grow faster. And so the dotted orange line shows our current global agricultural output. And then the solid orange line shows future projections. And then the green line is the productivity that we actually need to keep up with future demand. And so one promising way to increase crop yield is to increase the photosynthetic efficiency of plants. And given everything that I've told you tonight, can you think of a compartment that could perhaps be inserted into plants to turbocharge photosynthesis? So researchers are currently attempting to genetically engineer carboxysomes into plant chloroplasts. And so this approach, if successful, would be a great leap forward in the direction of higher yielding, faster growing crops. And just to show you that this is not a pipe dream and that researchers are getting close, here in that syringe is DNA that encodes for carboxysomes being inserted into the leaf of that plant. And then if we zoom in to the chloroplasts of those plant cells, and we zoom into those little black dots, if we purify those black dots away from chloroplasts, we find that they are assembled and fully functional carboxysomes. But even if we can do that, the question will always remain, would you eat carboxysome-powered wheat? So with that, a big thank you to museum staff who played a central role in the development of this exhibit and made our research accessible to a broad audience. Here are the main figures that put a significant effort into it. Thank you to the National Science Foundation that funded this exhibit, and to all of you for coming and supporting our research and outreach on microbes at the museum. Thanks for your attention.
Can everyone hear me? Is it working? Okay, cool. Hi, everyone. Thank you so much for coming. I'm Erica. I'm a PhD student in chemical engineering, so I'm not a microbiologist or an artist, sorry to tell you, but I consider myself microbiology adjacent, and you'll see that in my exhibit, the microbes themselves are the artists. So as April mentioned, I started the Microbial Masterpieces project at U of M with the aim of educating people about the importance of microbes to humanity. In this talk, I'm actually not going to go into that because Anthony did a really great job. So I'm just going to focus on my exhibit in the Natural History Museum, which is essentially a microbial ecosystem in a jar called a Winogradsky column. And I'll start off with some background on microbial ecology that's going to expand on what Anthony talked about a little bit. So Anthony focused a lot on the relationship between people and microbes, but it's not always about us, guys. There's also relationships between microbes. There are many kinds of microbe-microbe relationships, such as a mutualistic relationship in which both microbes benefit, for instance, by cross-feeding nutrients to each other. There's also competition. So microbes might be competing for different resources or for territory, and they might even actually kill each other by secreting antibiotics. We can have relationships in which one microbe benefits at the expense of the other, and some examples of that are predation and parasitism. And we can have relationships in which one microbe is harmed or benefited while the other is unaffected. And keep in mind, these relationships are fluid. So sometimes microbes are friends, sometimes they're enemies with each other, sometimes they're frenemies. It depends on nutrient availability and environmental conditions. So in nature, we usually, but not always, have many different species of microbes living together and interacting with each other and sometimes with hosts such as ourselves and plants. And things get way more complicated when you start to introduce abiotic factors into microbial ecosystems, such as the sun, water, oxygen, iron, sulfur, and a variety of other nutrients and inorganic compounds. So in other words, microbes affect each other, and they affect the environment, and the environment affects them. So where am I going with this? In microbiology, there are some species and strains of microbes that we are really, really good at growing all by themselves in a flask. And we call these pure cultures. But our limited understanding of microbial relationships in nature prevents us from growing and studying the remaining 99% of microbial species, which we call microbial dark matter, which we cannot grow in pure cultures. We know they exist because we have DNA sequences from the environment that tell us that they're there, but we just cannot grow them in pure cultures in the lab. And that's because they need certain conditions and nutrients that only their native ecosystems can provide, and we just don't know how to replicate those conditions. So how do we study them? Nowadays, we actually don't need to culture them as much anymore because we can use DNA sequencing techniques and other techniques. However, environmental microbiologists for these advancements in technology needed to study them. And that brings us to Sergei Winogradsky, the father of microbial ecology. He was a Ukrainian pianist turned plant scientist turned microbial ecologist turned farmer, and then microbial ecologist again. So a really talented guy who lived around the turn of the 20th century. And his whole idea was that microorganisms should be studied not in artificial laboratory cultures, but in their natural environments in competition with other bacteria. And that's a direct quote. Due to this belief, he left the lab, which was a weird thing for microbiologists to do at his time, and he studied microbes in their natural habitats, such as sulfur springs. And importantly, he also brought nature into the lab and conducted some of the first experiments in ecology, which had until then been largely an observational field. So one of the methods he developed to study microbes in the lab and to replicate their natural ecosystems in the lab was to build the eponymous Winogradsky column, in which he put water and mud from the environment into a glass column, he exposed it to sunlight, and he also added ingredients such as sulfur and carbohydrates to select for or enrich for species of microbes he was interested in studying. So over time, environmental gradients and interactions between the microbes, they lead to this stratification or layering of different colorful microbial communities. And so Winogradsky columns can turn out to be really beautiful and eye-catching. And for this reason, they're now not just used to study microbes, but also to educate people about microbial ecology. So in 2019, I saw a giant Winogradsky column at the Harvard Natural History Museum built by Scott Chmielewski. Maybe you're watching. I was with my friend Helen. Maybe you're also watching. Hi. And I really wanted to build one here at U of M. So I joined forces with Bruna Eunice Sanchez and Anna Urso, 2 other students who have since graduated from U of M. And we applied for and received the Experiments in Translation grant from Arts Engine. And with the support of Kathy Reister and Tom Bray from the Duderstadt Center, we got to work building a giant Winogradsky column. So I collected some mud from the U of M Arboretum where I used to work. Bruna and her dad built a stand, and we ordered a custom plexiglass tank. And then one very messy day, Kathy, my mom, and I mixed the mud together with eggs and shredded paper, and we poured it in the tank. It was a very messy day. And then we actually didn't see growth for a while, so we added some grow lights. There just wasn't enough sunlight reaching the column, so we added grow lights that you would normally use for plants to help the photosynthesizers get a head start. And after about a year in the Duderstadt Center, we moved the Winogradsky column to the Natural History Museum, where Melissa, Jade, Allison, and Todd helped me turn it into a museum-worthy exhibit. Thank you guys, and I'm sorry that I kept going over the word limit. Now you might notice that the column in this slide looks a little different than the last slide. Just go back really fast. So let's take a look at this transition through a time lapse that Tom Bray helped us set up. So there's going to be a little bit of flashing, and that's just like day and night changing. So the first thing I want you to notice is that in the top third of the panel, it's turning from black to green. And this is probably because the green cyanobacteria and the algae are producing oxygen through photosynthesis and killing off sulfate-reducing bacteria and archaea, which are black. And here you can see that there's all this orange sort of spreading down, and that's because some bacteria are oxidizing iron to derive energy, and they're essentially depositing rust around themselves. So the orange is like rust. And in these 2 areas, you can see the beginnings of purple bacteria growing. These might be purple sulfur bacteria, which do a form of photosynthesis in which they convert hydrogen sulfide to sulfur. And they get that hydrogen sulfide from the black sulfate-reducing bacteria. So let's take a look now at some macro photography shots taken by my brother in the audience. I put in some scale bars, but we took these like 2 years ago, so they might not be accurate, but they're around the right order of magnitude. So in this picture, you can see some very beautiful filamentous iron oxidizing bacteria. And here, in addition to iron oxidizing bacteria in the top right, there's some bright, deep orangish red. That could actually be purple bacteria, which aren't always purple despite their name. And while iron oxidizing bacteria create this orange color because they're depositing iron, purple bacteria have pigments called carotenoids that help them with photosynthesis, and those can actually be purple, red, or orange. In this picture, which I think is my favorite, we see some dark green, and those are probably green sulfur bacteria, and their green color is also from pigments that help them with photosynthesis. Just gonna let you appreciate this one because it's my favorite. And then here's another really good one where you have all these different community members present. So the main question I get when I talk to people about the Winogradsky panel is, why are the colors arranged like that? And I like this question because it's both artistic and scientific. We know that the colors come from certain pigments with the exception of the iron oxidizing microbes. We know that different microbes will grow better in different parts of the column due to the presence or absence of certain nutrients or due to the positive or negative relationships with different species of microbes. But honestly, the field of microbial ecology has a really long way to go before we can explain more than that. Like, why is there swirly textures in the bottom left? I don't know. Only now is technology catching up with Sergei Winogradsky's ambitions for the field of microbiology, and you could probably do 50 PhD theses just studying this particular column. So the Winogradsky column in my exhibit, it's not a complete exhibit or story, so I can't tell you exactly what's going on, but I hope that its beauty inspires people to be curious about microbes and their interactions with the environment, because this is a really exciting time to be in microbial ecology, and also a really important time to be in the field due to the climate crisis and the huge role that microbial ecosystems play in regulating global cycles of carbon and other important chemicals. But this exhibit and presentation obviously depends strongly on visuals, and that excludes a large portion of the population who are visually impaired, including more than 7 million Americans with a visual disability. It's really important that everyone has the opportunity to not just learn science, but also experience the wonder and joy that a beautiful piece of art like this Winogradsky column can inspire. So from the beginning of my project, my teammates and I were interested in developing the column to be more accessible. I'm running a little low on time, so I'm gonna skim over the rest. But this year I'm very grateful for accessibility interns at the Shapiro Design Lab for helping me create 3D printed tactile maps, which we're going to be reaching out to various community organizations soon that help the visually impaired community to see if we're going in a good direction with our tactile representations of the column, so that everyone has the opportunity to experience like the wonder and learn about the science of the Winogradsky column. So if anyone in the audience has any feedback about this, I would love to hear it afterwards. Or if you represent an organization that works with the visually impaired community, that would be great to hear from you too. So before I hand the mic over to Jim, I just want to say one last thing about Sergei Winogradsky. His work on microbial ecosystems helped him develop the ideas of sulfur and nitrogen cycles in nature, and this work led directly to the scientific idea of cycles of life in nature. And obviously a lot of human cultures around the world have an intuitive understanding of cycles of life and the interconnectedness of the world, which is reflected in a lot of ancient teachings. And Winogradsky's work validated this scientifically. He gave us a lot of methods to understand, quantify, and prove that everything on our planet is related in some way. And as Dr. Vecchiarelli talked about, understanding these relationships is key to protecting the planet. So with that, I'd like to thank you all for coming. Thank you to my family and my friends and to the museum, and I'd like to hand it over to Jim.
All right. I want to thank Amy for her generous support for my project and also for including me in this event tonight. And thank you all for being here. I came into this project knowing absolutely nothing about microorganisms, but jumping into the unknown disrupts habits and assumptions and offers new resources to me as an artist for my creative practice. We know that cultures develop and change through acts of integration, and so do people. In my public projects, I try to create unexpected encounters, and rather than grieving that I can't control every angle of lighting and every spectator posture and point of view, I've learned to turn porosity to advantage. Rather than demanding isolation from the rest of the world, as in the white cube of a gallery, I like to invite the world in all its ungovernable contingencies into the work, and thereby voice a tribute to the world that exists outside myself. My public projects bring attention to windows as membranes between interior and exterior spaces. They call attention to the interface of public and private identities associated with places and the people that use them. And they involve complex negotiations between personal vision and publicly situated entities. In doing something like this, I must account for the public spaces where people congregate to study and socialize, the flow of people from one space to another. That includes you, the intentions and assumptions of visitors, the disciplinary arenas invested in the study of what's collected here. But above all, I must take into account the architectural structures of the building, which is to say, I must study the site and make my work for that place. Site-specific art purposefully engages its context. Architecture, at its best, has always embraced this principle. But with the 20th century banishment of ornament from our public buildings, art became estranged from architecture. What we call site-specific art, however, acknowledges that an object's meaning is not something that it contains entirely unto itself, that it cannot be taken out of one context and put into another without altering its content. Site specificity embeds a work of art in a place. It expands and complicates the narrative of that place. It makes the site more permeable, more relatable, and it shifts our perceptions and expectations of it. My installations are designed for specific spaces and literally cannot be extricated from them. The vinyl can only be removed by destroying it. So my first task in doing my project here was to study the site. I constructed a scale elevation of the windows where I would install the project. I studied the primary structural and visual units. I took note of architectural features, columns, doors, corners, stairs, landings, banisters. I studied the light, where shadows might fall at different times of day. I assessed physical access for installation, and I thought about the movement of people through the spaces. Now, from the start, microorganisms were a thematic interest, but I really had no clue where to begin. Amy, I don't know if you knew that. How might I go about translating something like this Brachionis into a flat vinyl silhouette? My heart sank at the first samples I encountered. These are fascinating creatures, but how could I sustain visual engagement with such simple forms over such a vast space? So you see, vinyl comes in a limited range of flat colors. I can't use shading, for example, but I can suggest volume through contour and detail through interior negative spaces. The vinyl lies flat against the glass, but I can use implied overlap to create spatial dynamics. And I can use relative value contrast to make forms appear to advance or recede in space. Active contours of elements establish directionality and movement, and they frame the spaces between them. The framed views and the reflections on the open glass are ultimately what bring the installation to life. Columns and walls where shadows fall suggest further spatial and temporal dynamics as the sun doubles, shifts, stretches, and shrinks them. So I began searching for forms with what I call directionality to create visual movement and frame the negative spaces like this lacrimaria. Some had fascinating backstories like this rotifer and more familiar creatureliness like this tardigrade. Daphnia might work if I found a way to simplify them. They're certainly animated. They have these arms. But when I encountered the siliceous skeletons of polycystine radiolaria, my heart leapt with joy. You can't understand how happy I was. This I could work with. Acantharia, like elaborate jewelry. With some patience and attention to detail, diatoms might work, so exquisitely and delicately formed. Various algae had possibilities, Pediastrum, Microsterius, Xanthidium. I took special pleasure in being able to name them. I ultimately broadened my net to include the world of mites, So many mites, so little time, as one researcher put it. And many, many plankton, so strange and varied. I completed 350 ink paintings of the forms that I thought would be the most useful. I'm deeply indebted to the researchers, most of whom I've never met and who remain unknown to me. Their work contributed directly to the images on these windows. They opened my eyes to this amazing world. I worked with an undergraduate studio assistant, Sky Christoph, to translate the ink paintings into Adobe Illustrator vector files. This is my bank of digital images. A gathering of radiolaria, coccolithophores, echintheria, a few plankton that you see up there. Plankton because whether microscopic or simply small, they are a ready source of variety and action. Diatoms and various forms of algae for their complex, irregular geometries. So after months of arranging and rearranging my elements within the elevation, I arrived at a layout. The elevation allowed me to see the project space from a distance and devise ways to tie it together. Here, a group of radiolaria crossed the landing towards the stairway, crowned with microsterias and pediastrum. But they really only come to life on the windows, seen amongst shadows, reflections, and the glare of sunlight on the glass. Stairs are made for movement. Repetition and variation are essential for suggesting that movement. So there's a wheel-like form that eases you down from each landing, a motif constructed of a circular diatom behind a barnacle nauplius, a larva. It rolls into view more visibly at each stage of the descent, repetition and variation. On the largest landing, this structure illustrates a kind of typological game that's also played out on other windows. I'll use it as an example. In the black layer, in the back layer, are two giant annelids, huge worms, one facing up, one facing down. In the front layer, 8 tentacled creatures, hydra and schistosomes, kind of visually related by their tentacle-like appendages. And in the middle layer, a 4x4 square, a set of 4 diatoms and radiolaria, repeated 4 times in shifting order, ABCD, BCDA, CDAB, and DABC. Does that make sense to you? It's a Latin square. A Latin square is a fascinating visual pattern, but it's also an array that's used in combinatorics and experiment design, so I've learned. That logical arrangement is disrupted by a pattern of color relationships that doesn't correspond to the logic of the Latin square, which is further scrambled and intensified by cast shadows and shifting lights. Pattern making, pattern breaking. The complexity of living systems will always compel our imaginations more intensely than rigid, predictable hierarchies. That's at least my opinion. So I've divided the lower lobby glass wall into horizontal zones. Across the bottom is a pool of interlocking forms, deep enough to immerse the average viewer. Floating pairs of jellyfish divide the pool into subunits. The zone above the jellyfish is marked with lozenge shapes that are placed at a different set of intervals, diatom layered with radiolaria. And across the top, a large arc marks major intervals at corner, column, and the base of the stairs. Now, since ancient times, the tops of columns have been capped by floral and vegetative motifs. Lotus and papyrus in Egypt, acanthus in Greece. Why not a colony of E. coli paired with a diatom in Ann Arbor? Worms flank the building corner and the doorways. Demon guardians. The 3-part entry has been crowned with E. coli arches, a door to the Cathedral of Science. The roof ornaments on the Harold Washington Library in Chicago are shocking in their scale, but they have a long history in classical Greek acroterion, such as the palmette finial on the roof of the Parthenon. The museum now has its own microorganic acroterion. On the windows of the Natural History Museum, I'm using a commonplace commercial material to suggest a world of marvelous forms, normally unseen by most of us, though they permeate our world. Their exotic appearances, I have learned, are in fact evidence of how these organisms have adapted over time to very specific environments that have allowed them to survive, evolve, and prosper. Bringing them into artistic focus highlights what is inherent but hidden in all life forms, objects, and materials, most certainly in ourselves. Permeability and transience, what happens over time. This project also reminds me how museum display itself is never just facts, how it invites both looking at and looking through, how it is mediated by reflection, is open to countless interpretations, and combines unpredictable ingredients with theatrical fictionalizing. Thank you.
Thank you, Jim. This is, we're moving into the portion of the program now where we're going to be taking your questions. I was cheating and writing down some notes while we were listening to our panelists talk. I hope you were doing the same. I do have a couple of, before we get into our discussion though, I have a couple of just announcements and general information that might help folks. We do have, if you want to submit a written question, if you don't want to ask verbally yourself, that's totally fine. We have a couple of folks in the room who, if you want to raise your hand now, they can supply pen and paper, and you can submit your questions in that way, and somebody else will read them out. We also, of course, if you wish, there will be microphones available, and you can ask your question yourself. We want to be sure that everybody can hear. So please do, when you use the microphone, we say eat the mic in my line of work. Make sure you speak into it so that everyone can hear, both those in the room and everybody who's watching far away too. There is a Google form for everybody who is watching from far away. So please, if you're watching the live stream and you have questions, please do use this form to submit your questions for the panelists. We have a staff member who's monitoring those, and we'll try to balance questions from the live audience and the folks who are watching from far away as well. One more logistics thing, sorry, we just got to get you while the iron is hot. You won't be able to cut back through the museum if you parked in the Palmer parking structure. If you need some help navigating, one of the docents at the museum can help you figure out the path back to where you began from. They also have maps if you need it to help you on your way. So I'll try to remind you again about the parking situation as we finish the program. Thank you all. This was such fun to get to watch your brains work a little bit. And I also wanted to start this part of the program with a bit of an apology. I feel like especially people like Anthony and Erica spend their whole lives trying to educate all of us about microbes and their important role, and then people in my line of work systematically destroy public opinion about microbes by focusing on COVID and the other, some of the more nefarious bits of it. So sorry in advance or retroactively. Anthony, I have to ask you, there's a lot of places that we could get into this, but one of the things that you said just really stuck out. How is it that we have more microbial cells than human cells? I mean, how is that even possible?
Yeah.
Is it all in the gut?
It's 95% is in our large intestine.
Yeah.
And they're doing a lot of work for us to be able to get the nutrients that we need. And yeah, but I mean, so genetically, a while back, a few years ago, they thought that we actually had 10 times more bacteria in us and on us than human cells. But that number has been updated more recently, and it has been verified by other labs that it's roughly half and half.
To say there's more of them than us seems like an understatement. At the same time, Erica, your point about microbial dark matter and how much we don't know, was profoundly shocking to me. Do you want to say some more about the parameters of research that we have now?
Yeah, I can. Wait, I think I have to turn this on.
Oh, sorry.
Good. Well, first, microbial dark matter, it's a bit of a flashy term. It just means that there's a lot of microbial species that we don't know about, and it's estimated that there are like 99%, at least in bacteria. At least. Yeah, at least, yeah. So essentially what the field of microbiology has been doing for a long time, they try to create these pure cultures of microbes, and this is largely because microbiology has mostly been driven by medical microbiology, so there's a lot of interest in finding like this one kind of pathogen and growing it just by itself so that you can study it, and there's been less interest in just studying all the random microbes that live around us, which we now know are really important. But now we can study the microbes that are all around us without having to culture them, because there's a technique called pyrosequencing that essentially means that we can take DNA from the environment and we can sequence it really, really, really fast and then use bioinformatic tools to put all these sequences together. And each microbe has, I don't know how much detail I should go in, but each microbe, I guess, has a part of their DNA that it sort of is like an ID tag. So we can look at all these DNA sequences and we can see like what ID tags are there, and we can say, okay, these kinds of microbes are probably here in the environment. So we don't necessarily need to culture them anymore to know that they're there. Yeah. Does that make sense?
Yes.
Okay.
There's something that's peculiar to wildlife biology that you notice in years of reporting, and that's that the species of animals especially that are the most studied are the ones that people hunt or pests. Are there similar things about the kind of microbes that tend to get the most attention? That question is to either of you, Emily or Erica or Anthony.
I could start with the classic example of lab rat E. coli. E. coli is not the dominant microbe in our gut, but it is the microbe that the vast majority of microbiology labs have started with, very well studied. It is literally figuratively the lab rat of microbiologists. And the reason for that isn't, again, because of its importance in our gut per se. There are plenty other more important ones. It's because E. coli can grow very well in an undefined medium. For example, just putting together a yeast extract, for example, or a beef extract, that's, E. coli grew well in there, and that's why we started with it, and that's why we know so much about it.
Yeah, I'd like to add, we study a lot of pathogens, like microbes that make it sick, which makes sense. But as Dr. Becchiarelli talked about, microbes are really important for the environment and also for agriculture. And so, we've just been focusing on all like the bad pathogens for a really long time. And now I feel like there's kind of a renaissance in the field, especially thanks to these sequencing techniques where we can now go and appreciate and study the other types.
Yeah, it's a really exciting time to be a microbiologist now that you can study things that you don't necessarily have to grow in the lab.
Yeah. Jim, can I ask, would you better acquaint us with some of the microbes that you met as you worked on the project? Any that particularly caught your attention? You mentioned radiolaria, which...
I love radiolaria.
Such a
Aren't they
beauty.
beautiful? Should I turn this off?
I don't think so. I think we're good.
Okay.
You're good.
I love siliceous skeletons of radiolaria. Now, I'm going to misspeak, okay? So all you scientists out there, you can correct me later. Please not right now. So they're these skeletons made of silica. And radiolaria have those intricate structures. And they have been discovered in geologic layers going way, way, way back. So that means those delicate structures have been compressed in the earth over billions of years. And they can actually identify the age of layers, geological layers based on the radial area that are there, because they change over time. Now, isn't that amazing?
Yes.
And how does something with those intricate little openings, I guess that's why they call it polycystine, those intricate openings, how does that withstand the pressure, geologic pressure? So it must be engineered in some amazing way.
Jim, organic forms have been a fixture of your work going back a pretty long time. But was there anything about these forms that was different? Or, I mean, I definitely got the message that, you know, your work is so site-specific. A lot of the grist of it is just figuring out the installation. But was there anything about these particular forms that brought new ideas or challenged you that you hadn't run into before?
Absolutely. If you don't know anything about the microcosmos, you might look at them and say, boy, those are amazing abstractions. They're just abstract shapes. But of course they're not abstract. They come from a very specific life form. And I wanted what I put on the windows to have a specificity so that Erica and Anthony could say, so they wouldn't be scratching their heads too long. So,
Was there, I'm sorry to interrupt, did either of you say, hey, I know that guy, for any of the
Oh my gosh,
microbes that were?
so many of them I saw on the windows that I made going to the museum a part of getting an extra credit for my intro microbiology. There's a few students in the crowd right now that had to do that.
Good job, Jim. Good job.
they can thank me later. Yeah. So I came out of figure painting. And I was determined that this was not going to be distorted by the anthropocentric viewpoint. So how do I bring these creatures to life in a way that we as humans can project, empathize, want to know more about, without making them human-like? That was the challenge.
Yeah. I wanted to ask about something that's maybe a little bit more evident if you go and see Anthony's piece of the exhibition in the museum, but you got from the presentation that, you know, his work has to do with carbon sequestration in certain kinds of cyanobacteria. I'm at a place right now where literally anything that can offer some release on carbon is of great interest to me. Can you maybe give us a little bit more context about sort of where we are in understanding this process and what it would take to make it you know, one of the engines with which we meet the climate crisis?
Oh, okay. I can answer that. I'm trying to think of an answer that doesn't get too political. The short answer to your question is that we have to stop putting fossil fuels in the atmosphere. No matter what we do to try and fix this problem, it's not going to be fixed by these bandage approaches relative to the amount. It's a rate issue. We're putting way too much at a rate that any of these biological innovations can keep up with. So that's the number one thing, is to stop putting fossil fuels into our atmosphere, by far. And right? That was my son, Bowen, who put his hands up.
Yeah. Yeah, they right.
That's the number one way. The second is I do think that, you know, there's a lot of commercial innovations coming out. I don't think any one of them is going to be a silver bullet. I'm a fan of biologically inspired ways because, one, they are very efficient, as I presented today, and two, you know, as everybody might be seeing on the news, sargassum, you can make a ocean forest that's twice the size of the United States without us having to necessarily do anything. And so I think that it's understanding and harnessing the power of biology that might help us get us out of this mess.
From my reading of your work, it seems like to actually make a dent, with the understanding that it wouldn't be the only thing, the only process that humanity should rely on, isn't it something that would take an incredibly large amount of physical space just to accommodate the microbes that would do this work?
That's an excellent question. So if you think about it horizontally, yeah. But one of the reasons why algae and cyanobacteria are so useful is that you can build vertical bioreactors. And so as long as you're plumbing in carbon dioxide and they have an exposure to sunlight, there's no reason why you're limited to a pond. You can make three-dimensional piping, and so you can significantly increase the volume of what's sequestering carbon dioxide from our atmosphere.
And the output is sugar?
The output is sugar.
What are we going to do with all the sugar?
The cells.
Chocolate mousse.
I'm going somewhere with this. But I want to,
Yeah, I mean, I'm glad you brought that up because it's one of the things that I didn't anticipate when I watched, especially kids coming into my exhibit, is that they are blown away with the fact that this enzyme is taking, quite literally, atmospheric carbon dioxide and turning it into a sugar. And then that sugar is used, I mean, they don't give it out, they quickly use it for the cells to grow. But those cells that are now grown up can be used as mulch, as feed, for use to form a biofilm, or biofuel, sorry. Yeah.
I know that it's not a process that results in a pound bag of refined white sugar in the store, but my brain went straight to Kara Walker. There's this artist in Brooklyn named Kara Walker who did this absolutely astonishing, like 30-foot tall Sphinx-like figure. Had you seen a picture of this a couple of years back? It's called A Subtlety. And I just thought, microbes, this could be the engine of artistic practice in the future. Anyway, go look up Kara Walker on Instagram. She's amazing. Erica, I wanted to ask you a little bit more about the building process. Your presentation talked about building your Winogradsky. What did you call it? Like a slide?
Oh, yeah, I alternated between calling it a column and a panel.
Yeah, it is a column of sorts, right?
Yeah,
Even though
it's
not
generally built like in a cylinder, just like a graduated cylinder that you might have in lab. But because we got the grant and then the pandemic immediately started, and we realized people weren't going to be able to see it in person, so we switched to the panel so we'd be able to take pictures of it better.
Smart.
Yeah.
So we learned from your presentation that the ingredients, so to speak, are river water, eggs, and you fed it with paper?
Yes.
What kinds of paper?
Oh, okay. I know where you're going with this. Yeah. So I collected mud and water from the Huron River, and yeah, we just dumped in probably half a dozen raw eggs, including the shells, just mashed them up, and then we used shredded paper. But before I put the shredded paper in, at that point I was still very active on the Microbial Masterpieces Instagram, which I'm not so much anymore. But I had people write in like their hopes and dreams, and we wrote it down on the paper. We wrote their hopes and dreams down on the paper, and then put it into the panel as well. But then like weird things happened where some of the hopes and dreams like came true that were very like implausible. Just somebody lost their snake, and then they sent in like, oh, I hope I find my snake. So I wrote it down on the paper and I put it in, and then a couple months later they found their snake, which is very impressive because I lost a snake in my house when I was in 3rd grade, and we never found that snake.
Oh, wow.
Yeah.
Microbes manifesting.
Yeah.
I just want to point this out.
There's a whole nother line of work there. I
Yeah.
When you were showing me the column, you referred to it as a certain colloquial name. Would you care to share with the group?
It's my slime child.
Does
Yeah.
the child have a name?
Yeah, and we call him Sergei usually after Sergei Winogradsky, because calling it the Winogradsky panel and the Winogradsky column, it's just a lot of syllables. So we call him Sergei, and he's my slime child. And I feed him raw eggs.
I loved what you said about how there's so much we still don't know. For example, we know why the colors are the colors, but the swirls and the patterns. I mean, I guess my brain kind of went to, does that have to do with the organizational structure of how they manifest?
Yeah, so spatial organization is, I think, another hot topic in microbial ecology right now, where, it can just get very complicated. So one thing I want to make clear is I was referring to iron oxidizing bacteria, purple sulfur bacteria, green sulfur bacteria. So these are not like species. They're broad like groups of microbes, and in each of these groups there might be hundreds or even thousands of different species present. So when we talk about the relationships between all these species and how they interact, there's millions, trillions, I don't know, there's some exponential amount of relationships between the different kinds of microbes. And so probably some of the spatial organization is because microbes are, they're sort of icing out other microbes that might compete with them by secreting antibiotics or just taking up space. Or microbes might cluster together that have a mutualistic relationship. Or it might just be, there might just be a location in the panel where the nutrient conditions are just right for a bunch of different microbes. So they're all trying to, invade at the same time. But it really changes a lot over time. It's not static. They're always interacting with each other. Yeah, and some of the structures I think we can explain, for instance, the filamentous iron oxidizers. When iron oxidizers, when they oxidize iron, it sort of forms a sheath around them. So it's not, I think, that the microbes themselves are filamentous, although there are filamentous microbes, but they form these sheaths of rust around their bodies, and then they divide, and so then there's they divide by binary fission. They just sort of split in half, and then there's another microbe right next to where they started, and then that one also forms a sheath as they oxidize iron. So some of them we can explain, but there's so many different species in there that, yeah, there's so many projects that you could do. You have anything you want to add? Yeah.
I could seriously go on with these three all night, but I want to make sure that we're not overlooking questions in the audience. Shall we have a hand down here. Where'd the baby go?
Where's the baby?
So this is a question for, I guess, all four of you. In my field of science is rehabilitation medicine. And so in the COVID pandemic, people would come to the rehabilitation, which is the last stop before you go home, having survived the intensive care and multiple courses of antibiotics and steroids and other drugs. And their bodies were sterile. And we had to replenish their microbiome in order to get them nutritionally back to baseline and recover strength. So what's in your refrigerator? How do you feed your microbiome?
We take probiotics. Prebiotics are important to making sure you're eating the foods that your good microbes want to eat.
Right.
So, for example, fruits and vegetables and probiotics are useful. Things that are high in fiber, for example, are really good for your good bacteria. But you also bring up a really interesting point about the once you've destroyed your microbiome with a bout of antibiotics, there are ways of bringing them back with probiotics and yogurts and sauerkraut and kimchi and all those good things. The most severe things that can often happen, and although this is certainly not FDA approved, and it's probably not going to be FDA approved for any time soon, is in the most severe situations where people get hospital-acquired C. diff infections following antibiotic treatments, just like what was mentioned previous in Erica's talk, was that not only is bacteria useful in combating with each other, but simply just being there, taking up space, helps prevent warding off, or helps ward off predators or pathogens. Sorry. For the same reason why the best way to prevent weeds on your lawn is to just have lots and lots of grass, right? Nice dense grass. I told that to my class, and I realized that none of them really think about lawn care very much. but the best way to prevent weeds is to make sure that you have a lush grass, good density, and that's true for your gut as well. One of the ways to prevent pathogens from occupying your gut is just making sure that you have a rich microbiome that should be there. And so in these C. diff patients that have used all these antibiotics and now you have antibiotic resistant C. diff, you've essentially cleared it out, paved the way for them to grow without competition. What seems to be working with incredibly high efficiency is the repopulation technique.
So I can give you the medical term. It's called fecal transplant.
Fecal transplant.
Ew.
And it sounds awful, but it works.
But it works. It's remarkably efficient, and unfortunately, it's even to this day often given to point of no return patients or those that have severe C. diff, antibiotic-resistant C. diff. But there are companies out there that are trying to bottle it, that are trying to cap, golden poop,
Yep.
and so that's something for the future. Hopefully, can be approved because it's remarkably effective.
And that's sort of the theory. You have to feed the microbiome from above and below.
Right.
And
That's right.
so
And-
I would teach families how to make a smoothie at home with kefir and fruit and other prebiotics and probiotics and squirt it into the feeding tube. And that, of everything else we did in medicine, that was more helpful than anything for recovering patients and getting them home.
That's amazing to hear.
I'm reminded of Jim's Demon Guardians. Oh, yikes.
Sorry.
Other questions? Here's one.
Thank you all for, can you hear me? Yeah, okay, cool. Thank you all for 3 great talks. Erica, I know you and I have talked about this before, but since you didn't get to talk a little bit about the tactiles as much, whether I'd love to hear more about how you're exploring that and the different kinds of ways you are creating the variation that will allow people to quote unquote see or feel the masterpieces.
Oh,
Oh,
thank
this
you.
was the adaptive technology for accessibility?
Yeah,
Yeah.
yeah. Thank you for bringing that
wanna
up.
hear
I saw that the timer was at 9 minutes, and I panicked. So I'm very grateful for this opportunity to talk about this more. So I'm not working on this directly all by myself. So at the Shapiro Design Lab in the undergraduate library, I'm working with 2 accessibility interns, Alan Zhao and Daniel Ho, who have been really great. And so I've been bringing them pictures of my column, and they've been coming to the museum with me, and we've been looking at the different colors together and sort of trying to understand how to convey this in a non-visual way. So they are exploring different textures that could represent different colors. Also, the idea that maybe you could do some kind of elevation mapping to sort of represent the different shades or some combination. And then you could 3D print, this detailed sort of tactile translation of the panel. And so we were thinking of having a book where the first page is sort of January when, I first got the panel up, and then you flip, and then you can feel how the texture changes from January to February, and then you flip. And a different texture might correspond with a different microbe. However, this is a very early stage. They're printing prototypes, which is really great, but in the accessibility community, it's sort of don't do anything about us without us. So it's really important that as soon as possible we can get these prototypes to members who have visual impairments, whether they're blind or they're colorblind or have any other kind of visual disability, to make sure that we're going in a good direction. Also, I can't remember if I mentioned when I was talking about it before, we talked, there are plenty of people who have visual impairments who do work in microbiology, such as Dr. Mona Minkara, a professor at Northeastern University who gave us a lot of feedback early on in the project. She's a biochemist who gave us a lot of really good feedback, and she's really great because she's blind and she also works in biology. So yeah, it's still very much an early work in progress. And yeah, I'm trying to remember what I said. There's so much I planned to say. Yeah, still very much an early work in progress. We also at one point explored sort of like audio interpretations. That was very difficult. And we've thought about using textiles instead of 3D printing. So like it's soft, like softer, nicer textures to touch. But those won't be as easily sterilizable, which is still important. So definitely a lot of brainstorming, a lot of collaboration to do. But yeah, I'm very excited about that part, and I'm learning a lot that I wish I had known before about how to design for accessibility.
Erica, what were some of the things that came up in the feedback that as this trial and error process has been going on?
Well, we haven't had feedback for the 3D printed prototypes yet, but the feedback that Dr. Minkara gave me was trying to keep it simple, and also when you create sort of a translation for the visually impaired community, keep the visual elements in it because it should be still enriching for everyone. So that was a good point that she made. I sent her like the audio interpretations we made, and she was very supportive, but she did make it clear that they didn't make sense. So that's why we kind of went back to, we think the tactile version might make more sense. But we haven't had any feedback on those yet. Yeah.
Have you dabbled in scents?
In sense? Oh, I've thought about it. Yeah.
Oh,
okay.
wow.
So Sergey.
But what would that smell like?
It would
Depends
smell
on the
like rotten
bacteria.
eggs. Well, Sergey would smell a lot like rotten eggs
Sergey would
because
smell
I'm
like
literally pouring raw eggs in there. But I've thought about this a lot because, as you walk around in the world, there's a lot of smells that are microbial in nature. One of my favorite ones is called geosmin, and that's the smell, when it rains and there's like that particular earthy smell that comes up from the soil. That's from actinomycetes in the soil. They're making that smell, and I love it. I have a pair of earrings with like that molecule on it. So I thought, like, do we even need to necessarily like directly translate Sergey? The Winogradsky column, which I call Sergey. Do we need to necessarily translate it directly to a tactile representation, or can we evoke the same questions and sense of wonder through other senses, like smell? So I know you can get scratch and sniff, like rotten egg smell.
Yeah, right.
I was like, could we do that for a bunch of other cool smells as well? I don't know.
How about taste?
I love what you're saying because it's all about the importance of the imagination in making the science come alive. Just right there with your Winogradsky column.
That is something I wanted to ask about. It's evident that imagination, while we do have the technology now to see them, imagination has to play a role in our relationship with them, just because their whole world is, it's our world too, but their world is, different from ours in ways that we can't comprehend with the eyes. Jim,
I've got a question
sorry.
for them. Can I have a question? Yeah. So there's a point where these complex forms erupted into my world. And I went, whoa, is that a single cell? How can a single cell have this complexity to it? And then others, no, that's not a single cell, that's multi-cell, surely. And then I ran into, in my reading, I ran into the thought that the complexity of the forms that were setting off triggers in my imagination were a result of the evolution of the microcosmos, the denizens of the microcosmos, where they became more specialized, occupying different ecological niches. I'd love to hear more about that. Like, am I just making this up?
No, not at all. One of my favorite reviews that talks about bacterial and prokaryotic cell morphology starts with the sentence that most people could care less about the morphology of a cell, and that's a shame because it seems as though bacteria care very much. And
Yeah.
And yes, it's definitely evolutionary based. Of course, it's encoded in their genomes, and it's there for a reason. But unfortunately, to your point, I mean, in my class, I teach how a spherical bacterium may have genetic components that will turn that sphere into a rod, and that's largely where the field is at.
Wow.
And so,
So I'm way ahead of the curve.
You're just light years ahead, and it's an active area of research. Like, what are the genetic determinants that allow a sphere to go to a rod, and how does that rod turn into something that's more filamentous? And then, what is the genetic determinant that allows for branching of that filament? That's where we're at, and obviously there's an evolutionary requirement for them to have these beautiful morphologies, but how it's done is still complete open territory of research.
So you used the word prokaryote, and I ran up against it, and eukaryotes. Would you kind of give us, there may be one or two out there that don't follow that, but I certainly don't. Could you?
Sure. Eukaryotic cell, eukaryotic organism has cells that have a nucleus. A nucleus is a membrane-bound organelle that has the genetic information within chromosomes. And prokaryotes are those that don't have a nucleus. So bacteria is one domain of life that's considered a prokaryote. And the other major domain of life is called archaea. And that is the next frontier of understanding prokaryotes, because they're actually a mixed bag of eukaryotic features and bacterial features. And so understanding the connections between eukaryotes, bacteria, and archaea, which are prokaryotes, will help us better understand how these three domains of life diverge from one another.
I feel like I'm dragging us back to biology
Yeah,
100, but how does it not have, how can some of them not have a nucleus?
Well, I mean, life first, the vast majority of life, the right answer, the right question is why do you have a nucleus? The vast majority of how, the vast majority of life originated as bacterial, and the vast majority of life by far is bacterial, and so the vast majority of life doesn't have a nucleus. A nucleus eventually evolved as an organelle. And it's thought that these membrane-bound organelles that our complicated cells have occurred through what's called endosymbiotic theory, meaning that one day a cell was engulfed by another cell, and instead of eating that cell for nutrients, it said, hey, that cell actually could be useful if I keep it around. So that's how an ancestor of cyanobacteria became a chloroplast. And that's how a bacterium eventually became our mitochondria, which are the powerhouses of the cell.
Right.
And so it was these relationships.
It's amazing.
Come on, I do remember that much.
Sorry,
from
I think I'll stop.
biology.
Yeah, this is turning into
No, I love it.
I think the laughter was that the mitochondria is the powerhouse of the cell, because it's drilled into our heads. There's a lot of memes about it.
Science last ones. Okay.
I don't know much about the mitochondria other than it was a bacterium.
We've got a question in the back.
So we have a couple questions coming on from our live stream audience, and this one is for all panelists, maybe to pull us back to the art side of things a little bit. Scientists typically try to make their thought processes objective and generally avoid words like beauty in formal scientific discourse. But how might scientists actually benefit from embracing artistic thought?
Oof. Take it. Go for it.
Okay.
She just did it.
Can I do it? Can I do a little? Well, go ahead. Sorry, I have possible reframe, but I want to hear what you think.
Okay, well, I think, when we formally talk about science, we do refrain from using words like beautiful and wonder, but I can tell you, as somebody in a lab with scientists all the time, every day, we definitely experience it and we talk about it openly. My friend Valentina, sometimes I walk into her lab and she's like covered with blood, and she tells me about this beautiful umbilical cord she just dissected, and she's like, it was such a beautiful umbilical cord, and we get excited about it together. So we do talk about beauty. We do talk about these things that inspire us and sort of spark joy in us on a day-to-day basis. And it's acknowledged among us. I think when scientists talk to the public, we try to appear as though we are purely objective. And the scientific method is objective, but scientists are human, so we certainly are we are definitely very driven by beautiful things that inspire us to keep asking questions.
Would it be possible to fully comprehend the world of microbes unless you have the creative imagination for how their world works? The artistic imagination?
I mean, for me, I'm a cell biologist, so for when we study microbes, we are actually looking at them. And so, I am certainly inspired and seeing the different things that bacteria can do, but we quite literally look at them and how they're functioning, both on the outside as well as peering inside them.
But bacteria have to communicate with each other. There's information transfer. So how far does that information transfer reach? Is it reaching you?
Oh, interesting. Oh my gosh.
Well, certainly, yeah, I guess.
Sorry, go ahead.
Well, they would communicate information to each other by sort of passing chemicals to each other. That definitely affects us, and it comes to us. So I think, correct me if I'm wrong, but about like 80% of serotonin, which is one of the hormones that makes us happy, is produced in our gut by bacteria. Please fact check me on this, but
No, that's right.
yeah, okay. Yeah, and so that could be something that you might call like microbial person communication, because there's all these microbes in our gut producing serotonin that then somehow makes its way up to our brain and makes us happy.
The way I took the question was one of by like looking at these bacteria and generating
that's-
data.
Both of them are great approaches. Keep going, keep going.
With me. Well, there are some miraculous things, remarkable things that you see, especially, so I mean, this sort of diverges from the question, but one of the best things about science, especially when you're a student or when you're a researcher, is that especially as a cell biologist, when you're in this pitch black microscopy room, there's nobody there. Sometimes you're there at 2 o'clock at night, experiments are not working, and then one day, nature unveils a piece of it that no one on Earth, maybe the universe, has ever seen before. And I can't think of a more spiritual experience than that eureka moment. I am jealous of my students and postdocs all the time now, because that's one of the moments that I miss the most of being at the bench. So in that vein, absolutely.
Yeah. So in a way, it is reaching you. It's not like having a chat with you, but
I mean, there's certainly times in which we don't even know what is the question that we're trying to answer. And by looking at these bacteria under the microscope or doing biochemical experiments, they're slowly telling us something about nature. And it's only by finally putting all those pieces of data together and doing the right controls that we unveil this part of nature. So in that way, they are communicating, maybe indirectly.
But in your narratives, you already demonstrated that by observing this world, these microbes within their environment rather than in a petri dish, that you're learning more about them. Isn't that right? Yeah.
Yeah, you can answer different kinds of questions. I think there's still definitely a lot of value of growing things in a petri dish, but we're starting to look into the questions that we can answer outside the petri dish. I think to circle back to the original question is like how can beauty sort of maybe enhance science? One of the things I was thinking about was my 6th grade geology teacher was always trying to get us to draw more, because he was like, oh, observation, like drawing forces you to observe, and so that forces you to learn about things. So I think that taking that can certainly be a lesson for all scientists. Is sometimes we have this like one hypothesis and we just follow it single-mindedly, and we might miss interesting things that are happening in our experiments that aren't necessarily what we're initially focused on. So I think remembering to take the time and observe things really closely, whether that is through looking at something closely and drawing it, or by listening or using all of your other senses, I think that it would make us all better scientists.
That's a great answer.
If the weather improves, I'm looking forward to coming down with my sketchbook and and taking a run at Jim's forms.
No, go to the originals.
Okay, that too. But it's so much nicer outside. Any other in-person questions, or we could definitely take another from the online audience. Maybe one more. We're getting close to the end here. We do have one down here, toward the front.
Straightforward one.
Oh, I'm sorry. There's someone in the back with the mic. My bad.
Yeah, I'm wondering.
We'll be here. We'll be here.
If you imagine, do you imagine what microbes might be if they lived elsewhere in the solar system?
Oh, such a good question.
Did everybody hear that? Yeah. Who wants to take a swing at that one?
Oh my gosh.
They already look like remarkable life forms from another planet, right?
Yeah, so I mean, there are researchers from NASA and also throughout academia that are looking at extremophiles. Extremophiles are microbes that grow in conditions that a human would consider normal, outside of normal. So, you know, outside of 1 atmosphere of pressure, outside of a salt solution that would be similar to the saline solution of an IV bag, outside of what we would consider to be a nice temperature. So falling outside of those ranges, let's think about temperature, for example. You know, there are bacteria on Earth that can grow and not just survive, but thrive at 0 degrees Celsius and also at 120 degrees Celsius. Those are the microbes that are going to be probably closest to the type of microbes that we would see on other planets, because they will be living in what we would consider extreme environments. And so the type of bacteria that we see in like Mono Lake in California, Yellowstone National Park, that's a really useful reservoir of the types of microbes that we might be able to see in other planets. For example, in one of those lakes, there was a bacterium that was identified that can eat arsenic. And so, although we don't know for sure, of course, because we haven't directly identified one from another planet, if they look like anything, they'll look like extremophiles.
I want to thank everyone who came out tonight at the end of their week, and of course our panel. I want to thank everybody at the museum who made this exchange possible tonight, and everybody who's part of the digital audience too. We really appreciate you. We have been reminded tonight of the inseparable nature of the world that we see and the world that we cannot see unaided. And we've expanded our definitions of community and ecology and microbe, and hopefully gotten a little bit better acquainted with what those words can mean. We've heard perspectives on how we're sharing the connectedness through art and through design. And I just wanted to share a couple of parting thoughts inspired by the University of Wisconsin Arboretum. And this is a thing, the Microbes and Social Equity Working Group. As we consider our impacts on the planet and the fate of human societies, we must also consider our impacts on microbial systems with the same gravity. Microbial ecologists at the University of Michigan and throughout the country and across the world are coming together to explore how our understanding of microbial systems has been shaped by our biases and our borders. Microbiologists are asking big questions. Who gets to study microbiology and where? Which microbes get studied, and how are those studies influenced by systems of oppression? Any ethics we hold about the environment and how to care for it, about social justice and indigenous sovereignty, about science communication and the power of art must be extended to the microverse as well. The artistic endeavors that we've seen tonight are just one avenue for creating some accessibility around these things and equity in our study of the unseen world. It may sound like counterculture, but truly the future depends on it. Thank you, Anthony and Erica and Jim, and thank you all for being here tonight. Can we please give our panel a round of applause?